Evaluation of DNA technology. Genetic engineering undoubtedly brings many benefits to mankind, but it is not without its risks. It is therefore important to evaluate the ethical, moral and social issues around this issue. The benefits of recombinant DNA technology. Microorganisms can be modified to produce a range of substances, for example antibiotics, hormones and enzymes, that are used to treat diseases and disorders. Microbes can be used to control pollution, for example to break up and digest oil slicks or destroy harmful gases released from factories. Care needs to be taken to ensure that such bacteria do not destroy oil in places where it is required, for example in car engines. To do this, a suicide gene can be incorporated that causes the bacteria to destroy themselves once the oil slick has been digested. Genetically modified plants can be transformed to produce a specific substance in a particular organ of the plant. These organs can then be harvested and desired substance extracted. If a drug is involved, the process is called plant farming. One promising application of this technique is in combating disease. This involves the production of plants that manufacture antibodies to pathogens and the toxins they produce. Alternatively, the plant can be modified to manufacture antigens which, when injected into humans, include natural antibody production. Genetically modified crops can be engineered to have financial and environmental advantages. These include making plants more tolerant to environmental extremes, for example able to survive drought, cold, heat, salt, or polluted soils. This permits crops to be grown commercially in places where they do not grow at present. Globally, each year an area of land equal to half the United Kingdom becomes unfit for normal crops because of increases in salt concentrations in the soil. Growing of genetically modified plants such as salt-tolerant tomatoes could bring this land back into productivity in a world where millions lack a basic nutritious diet and with a predicted 90 million more mouths to feed by 2025. Genetically modified crops can help prevent certain diseases. A type of rice, called golden rice, can have a gene for vitamin A production added to it. This vitamin A can help reduce blindness. Genetically modified animals are able to produce expensive drugs, antibiotics, hormones, and enzymes relatively cheaply. Replacing defective genes in gene therapy might be used to cure certain genetic disorders such as cystic fibrosis. Genetic fingerprinting can be used in forensic science. The risks of recombinant DNA technology. It is impossible to predict with complete accuracy what ecological consequences will be of releasing genetically engineered organisms into the environment. The delicate balance that exists in any habitat may be irreversibly damaged by the introduction of organisms with engineered genes. There is often no going back once an organism has been released, although suicide genes can be inserted or the organism engineered so it can only survive when a supplement is added. A recombinant gene may pass from the organism it was placed in to a completely different one. We know, for example, that viruses can transfer genes from one organism to another. What if a virus was to transfer genes for herbicide resistance and vigorous growth from a crop plant to a weed, and then it outcompeted the crop plant? What if the same gene were transferred to pollen to other plants? How would we be able to control the weeds? Any manipulation of the DNA of a cell will have consequences for the metabolic pathways within that cell. We cannot be sure until after the event that unforeseen byproducts of the change might be produced. Could these lead to metabolic malfunctions, cause cancer, or a new form of disease? Genetically modified bacteria often have antibiotic resistance marker genes added. These bacteria might spread antibiotic resistance to harmful bacteria. All genes mutate. What then might be the consequences of our engineered gene mutating? Could it turn the organism into a pathogen? What will be the long-term consequences of introducing new gene combinations? We cannot be certain of the effect on the future evolution of organisms by that gene. Will the artificial selection of desired genes reduce the genetic variety that is so essential to evolution? What might be the financial consequences of developing plants and animals to grow in new regions? Developing bananas, which grow in Britain, could have disastrous consequences for the Caribbean economies that rely heavily on this crop. How can we take the technique of replacing defective genes? It may be acceptable to replace a defective gene to cure cystic fibrosis, but is it acceptable to introduce genes for intelligence, more muscular bodies, cosmetic improvements, or different facial features? Will knowledge of and ability to change human genes lead to eugenics, whereby selection of genes leads to a means of selecting one race rather than another? What will be the consequences of the ability to manipulate genes getting into the wrong hands? Will unscrupulous individuals, groups, or governments use this power to achieve political goals, control, opposition, or gain ultimate power? Is the financial cost of recombinant DNA technology justified, or would the money be better spent fighting hunger and poverty that are much the cause of human misery? Will sophisticated treatments, with their more high-profile images, be put before the everyday treatment of rheumatoid arthritis or hemorrhoids? Will such treatments only be within the financial reach of the better off? Genetic fingerprinting, with its ability to identify individuals' DNA accurately, is a highly reliable forensic tool. How easy would it be for someone to exchange a DNA sample maliciously? Is it immoral to tamper with genes at all? Should we let nature take its own course in its own time? How do we deal with these issues surrounding the Human Genome Project? Is it right that an individual or company can patent and therefore effectively own a gene?